Now we have models like in vitro, MatTech and Epithelix have now got the nasal, the conducting and the alveolar. We've got disease models. So many different test systems out there that we can now start to answer different questions related to different parts of that complex organ.
Hello and welcome to the platform of conversations where we bring experts from different healthcare domains to share their insightful thoughts over here. And today I am Rohini Kanbarkar and I'm pleased to be joined by Dr. Clive Roper, a leading expert in vitro toxicology, advanced 3D models and new approach methodologies (NAMs). Dr. Roper has spent over 2 decades leading the global programs in the skin absorption, regulatory in vitro toxicology, advanced tissue models and respiratory toxicology, as well as the director of in vitro toxicology. And in 2021 especially, he founded Roper Toxicology Consulting, supporting the biotech, CROs and the global organizations in the development, regulatory acceptance and the applications of NAMs. So his work has been cited also in OECD test guidelines, and he continues to advise the regulators and the scientific bodies worldwide. And it's my pleasure to have you on this podcast. Welcome, sir.
Thank you very much, and I'm delighted to be here with you.
Yes, same here. So when we talk about your work, you have been working on the organoids, 3D cell culture and everything. Can you give us a brief about what these things are and how important it is in today's generation?
Yeah. Well, quite often people will ask me, which test systems should we use? They'll say, should we use organoids? Should we use spheroids? Should we use primary cells? Should we use cell lines or iPSCs? And I say to them, well, what you really need to do is to tell me why you're doing the experiment. So what is your context of use? What are you trying to answer? And also, what have you got access to? So you may not be able to access certain types of cells, or they may not be appropriate. And you may end up finding that you end up with a mixture of organoids, spheroids, primaries, iPSCs. But you're trying to make sure that your test system reflects the question you're trying to answer, and your test method that you develop around that answers the question. And you'll find that if once you understand what you're trying to answer, you can then start to build together the test method and test system, which will quite often be a surprise to you of what you need. But often it's also what have you got access to? And sometimes you might need to actually say, we don't have the things that we want to generate, but we have access to this. And maybe we can answer a slightly different scientific question.
Yes. So we need to first answer that question, like what do we want out of this?
Decide what your question is. Decide. So if you wanted to know what inhalation toxicology is, well, you've got to decide: is it the nose?
Okay.
Is it the conducting?
Tract.
Is it the actual respiratory alveoli? And once you can do that, you can then start to answer those questions. Do you know what the adverse outcome pathway is? So what's the toxicology you're trying to do? What are the mechanisms? Once you know those, you can start to answer the questions.
Yes, I mean, understandable. And when we talk about organoids and microphysiological systems, which are gaining visibilities nowadays across all the industries, I would say. So from your experience and from your perspective, what determines whether these models become true decision-making tools in the industry?
If you're looking at a discovery question, you can use any tool that you want. You just have to have confidence that it's going to answer your question. If you then want to take it further and you want to actually use it in a regulatory process, so we're moving from that decision-making to regulatory, then you really need to get more information. And at the moment, the regulator's really very interested in seeing these data. They know that many companies are using these in vitro models to make decisions, and then often they will then go ahead and go through their standard safety assessment program with rodents, with animals, and then they'll submit. So they never see the decision making that's been going on, and they know they are. So what they want to say is, so why don't you submit that data too? Why don't you come to us? But what they want you to do is when you've made those decisions, use it as a decision-making platform, you've gained some evidence that gives you confidence.
Yes.
So you've seen perhaps that it replicates some clinical data from a known compound, which has clinical results. It represents a negative in clinical data that you know there. So failed drugs, for example.
Yeah.
And once you've got that information, you can then submit that alongside that information, your compound's information. The regulators are much more likely to take it on board.
Exactly. And now that you spoke about failed experiments, right? And there are always some kind of loopholes or gaps in the research that we are doing, or maybe the project that we are doing. So what do you think, what scientific or validation gap still limits the regulatory confidence in advanced 3D cell cultures?
Well, we are often asked to have validated test methods, and we do have a lot of problems with that. So if you want to validate, you might say, well, we've got lots of animal data, in vivo animal data.
Yes.
But we know that in many cases, those in vivo studies aren't replicating the human.
Yes.
So, it's well said, it's been said many times that 90% of drugs that go into a clinical trial fail. Now, maybe something like 60% of those failures are due to preclinical testing. So we can see that it's not the animals are not predicting what's happening in the humans. There are obviously many other factors that fails a drug. So if we can incorporate the reasons why we don't want to use an existing test and demonstrate the strengths and weaknesses of our assays, then and the strengths and weaknesses of the current in vivo tests, again, the regulators will look at that.
Okay.
So what they really want to see is evidence that they can have some confidence. They want to look at reliability, reproducibility.
Yes.
And harmonization itself, standardization and harmonization doesn't mean to say we will use one chip and we will follow one method. What it says is that it's got to have certain quality characteristics. So you've got there's a lot of work going on in drug-induced liver injury at the moment, and iStand program has Emulate Bio.
Okay.
But we're also running the 3 Hours Collaborative. I'm a member of the 3 Hours Collaborative. We are also running a program, multiple labs in drug-induced liver injury, and we're looking at those quality processes. So things like albumin production, urea production. So in standardization, you're not looking at having everybody do the Emulate method. There's nothing wrong with the Emulate method. I'm just saying that's going through that process. What we're saying is that you can use all these other technologies because they have the same quality criteria in place.
Yes.
So that gives us different technologies to use. And in very complex disease models, we want more to test because they each have some of them will be better for different classes of compounds, for example. So the more assays that we can test to take out drug-induced liver injury, the better it is.
Yes, and we'll have perspectives, different spectrum of results.
And we don't end up with everyone just being forced to follow one route, because the other problem with that is that when we have improvements, we have to go and start again.
Exactly.
Whereas really what we want to be able to do is say that we've still got the quality parameters in place, but we now have a better assay because we've made developments over months and years.
Yes. And when we talk about modalities or the emerging modalities, basically, the inhaled or dermal products that are present with unique safety challenges, right? So where do NAMs offer the most immediate and measurable impact today?
So like inhalation there's so many opportunities. We have so many test systems out there. When I started working in inhalation toxicology, we had mucilaire only.
Okay.
So that's nasal mucilaire. Now we have models like in-vitrolize. We have the in-vitrolize model. We have the MatTech and Epithelix have now got models of the nasal, the conducting, and the alveolar. We've got disease models. We've got immune lung and macrophage models. So there's so many different test systems out there that we can now start to answer different questions related to different parts of that complex organ. So it's a multi-organ. The lung is multi-organ. Here's the first one. We finished with the alveolar tissues. We've also got a vast number of technologies out there, so we can measure simple toxicity by MTT, for example.
Yes.
But we can also look at cytokine release. We can look at cytokine storms. We can use huge amounts of immunology. So we can use lots of pathology. We've got many different techniques, many different stains that we can use. So you can use all of these technologies, and a lot of them are used in current animal tests. They transfer nicely into the in vitro tests. A lot of them are clinical tests. Again, they will transfer into these models. So there's a huge number of tests that we can be utilizing. We just need to have imagination to make sure that we build the design to answer the question based on an adverse outcome pathway, the mechanism, and that we can build all of the mechanisms in place that relate to that human disease. Okay. So there's a lot in place. They're not going to be a single study which will answer the question. It's going to be batteries and different layers. And I think we talk about this a lot in a paper that we published in 2023, where we looked at generating testing for commercially available tests in the liver, the lung, cardiac, etc. So people can look at that as well. But definitely it gives us the idea that we can actually use many commercially available systems to generate huge amounts of toxicology information. We don't need to go near the animals. Exactly. That will come later. We still requirements, but what we can use is a huge amounts of development and complex toxicology from these other models. We then do our safety battery, and eventually we will know how much of that data we're generating in the question and answering those scientific questions, and then we gives them more confidence to start replacing those animals.
Okay. And we can analyze the difference between
and having a difference between a rodent or an animal in vivo test and a human in vitro is not a problem. We used to have a problem with it. We used to be going, we're not predicting the in vivo.
Yes.
And actually what we didn't say was, we're comparing in vitro, in vivo, we would say the in vitro system fails. We always forgot it's a species difference.
Yes.
So we really need to understand that when we're making these transitions, we've got to understand that there's going to be a species difference.
Of course.
And increasingly now we have animal in vitro models that we can use. We can use those for protecting animals. We can be using those for translation. And of course, people often forget we're always talking about human relevant or human. Well, what about the veterinary pharmaceuticals?
Exactly.
Their clinical species is the pig, is the cow, is the dog.
It's a cat and everything.
So So and they are not, and that actually that gives us a real problem when we talk about animal free. Yes. What we need to be really thinking about perhaps more is in vivo free.
All right.
And trying to say that yes, those animal in vitro models are important because we can use them to protect animals, because we're still going to be using animals for a long time.
Definitely.
So we can protect those animals by doing in vitro assays. We can get information that might say help with the dose range finding. So we might choose do a lower or a higher dose range finding. We might decide that there are certain toxicities we've got to look out for, bear in mind to look out for those better. So we're really giving welfare information.
Yes.
We might show, when we run in parallel our human in vitro assays, that we see that they're not a good model. And in that case, we might say we need to choose a different animal, or it helps us to justify why we should be using an in vitro only answering question.
And then how can these NAMs and the human relevant models that we are using will help to reduce the late stage attrition and they will help to improve the predictability of the drug development?
We can't just go ahead and just throw any test system together. We have to understand what we're generating, is that reflecting the human disease? Is that reflecting, are we generating efficacy data based on the same mechanism that occurs in the human? Frequently our efficacy in animals fails because we will have, for example, a very, very fast acting chemical causes the observed disease. Yes. We then treat that animal, we find a response, we find a drug, we put that into a clinic and it fails. And that's because the human disease has often taken many years to develop. So Alzheimer's disease, you can produce in animals very quickly. In humans, it takes 10 decades and decades. Yeah. So if we can use tissues from Alzheimer's patients, cancer, the same thing. You can create them in animals very quickly, but the patients have generated. So what we need to do is make sure that our models really reflect and really have the same mechanistic basis of the humans. And I think that's what we're seeing increasingly, people understanding the mechanisms, building them up with multiple tests to answer a question, instead of saying, we need an animal, it's complicated, we don't have that with an in vitro system. We don't need to. We're building the mechanisms. So we look at the adverse outcome pathway. We understand all the mechanisms. And a great example is skin sensitization. So there are multiple mechanisms. We can now predict it very well with doing 3 assays that pick different key events. So the more that we use examples like that and build examples like that, we'll become much more strongly evidenced that we have the mechanism, and that's why it's a superior model.
And when having advised multiple regulatory agencies which are there, what signals suggest regulators are increasingly open to the NAM-enabled decision making?
So there's a lot of things. They go to conferences, they see the new technologies, and they talk to people, so they know they're there. They know that people are using these in their development programs. Yes. And then suddenly they don't see it in the submissions, and they actually want to see them. They know that that they're the final gatekeepers to protect those clinical trials.
Exactly.
So they know that there's going to be a very high chance that any new clinical trial will fail. And they're looking at those technologies. They know that we've been doing those tests in animals. In the US, it was the 1938 Act that says you must do efficacy and safety in animals. Well, it was in 2022 that they changed it to say animals plus in vitro, organoids, computational. Yes. And so they know that these technologies are there, and they know that a lot of them are superior. What they want to know is, what confidence have we got in that model? How does it compare with the current method? And if you can demonstrate the strengths versus the weaknesses of your strengths and weaknesses of your in vitro model or your computational model versus the existing animal test, then they're very interested. And they want to learn. And if you take FDA, they're working. They have a memorandum of understanding with the NIH. The NIH has been working very strongly with the EPA for many years. Okay. So there's a lot of experience in sort of starting to understand that. And I've certainly taken quite a few materials, quite a few programs through with companies through these different agencies. And it's about demonstrating that this assay does what we need to do to protect human health.
So there are a lot of partnerships that you have seen in between the industry, academia, and collaborations that you have seen.
Yeah.
So now that we are talking about collaborations, so sometimes the platform-based approaches are becoming central to the drug development and the biologics part also. So how can researchers who are involved in the NAMs and 3D model platforms accelerate the validation, standardization, and the regulatory acceptance towards this?
So I think I've said some of the things there. So, they need to demonstrate that reliability. Yeah. So the reliability can come in many forms. It could be repeatability, which is running the same assay. It comes from analytical chemistry, actually. So repeatability is you run the same the same batch on the same day repeated, and you see that there's no variability. And then you then look at on different days.
Yes.
And so you're looking at, with different batches, do you still get the same answer, even though using different batches? That could be different batches of cells.
Like the reproducibility of
and
it.
that reproducibility. So if you see that they fulfill your criteria for acceptance, that data can then be demonstrated to the agency to show that there is that reliability and robustness. One of the problems that we often find now is that we have very interesting models, but we still have a lot of variability. And people try to improve their models, and they haven't actually realized that the problem is that it's their media that's the problem. So many people use matrigel. It's a mouse sarcoma, so it's not nice. But it's also highly batch variable. We don't know what it's made up of. And what it's giving us is an assay that is related to the variability of the media, not the variability of the assay. So what we're now doing is we're generating lots of new models, lots of new media that are animal-free and chemically defined. So I work with a company, I'm part of a company called Pvt Matrix.
Okay.
We produce these materials. Within the NC3Rs, I'm on the board of the NC3Rs. We have a lovely page. Please go and look it up. NC3Rs scaffold, it comes up. And on there's a whole list of companies who produce animal-free, chemically defined media. Now, once you've fiddled around with them, and it might well be you use 3 or 4 different suppliers, because remember, matrigel is made up of hundreds of chemicals from the animal.
Yes.
So you don't just necessarily use one animal-free, chemically defined product. Mix and match. All of those different categories will then help you, because you've always got to optimize. Use those. We'll help you with those optimization. You don't need to use matrigel.
Like tick all the combinations.
And there's other companies who are working to produce end bovine serum albumin and to end fetal calf serum. So they're all the same problems. They are biologically very variable because it's all the batches. Even the human derived media have a problem because we're all different.
Exactly.
So you can't keep taking my blood to make media. But of course, if you can remove that variability, now we can really optimize the tests, and we can use those to improve the reliability of our tests.
So what we need is the right approach, and to the right people, I would say, the right partnership and the collaboration.
It's all about stakeholders. Make sure your stakeholders are there. So look at the people that, so in the 3Rs Collaborative, we have a great site, MPS hub, and on there we have lots and lots of MPS companies. We also have been generating a new group, which is enablers, and they've included consultants now. But those enablers will be companies who make, for example, plasticware, or they make the chips, or they make organoids, or they supply cell lines. All of these companies together enable us to develop more reliable models, and so look out there. So do go onto the 3Rs Collaborative MPS hub.
So when we are talking about these partnerships and collaborations, what role can the global platforms and conferences, one like the 3DCC 2026, play in aligning these stakeholders and accelerating next generation in the safety science?
Well, I mean, I think we're doing the right thing already. We've got a very and a really exciting lineup, and unfortunately I'm missing a lecture now to be here with you, but of course I'm happy. But we've got some fabulous posters. I've been around and started looking some posters, and we're going to be doing a poster award. So SDIV is sponsoring a poster award, so I'll be giving that. Now, we start by learning, and the people who start there are the people who are doing the posters, and tomorrow they're the people who are doing the lectures. So this is a great time for students to learn from a lot of expertise. It's also us older experts. We're learning a lot from those students. So they're doing really exciting projects. But it's about learning. But don't be afraid as a student to ask one of the older boys and girls questions. You know, we had great mentors. We want to be good mentors for you. So
yeah, it will be a win-win situation.
we love doing it. You know, we're not going to be here forever. And
Yes.
and the best way that the great work continues is by mentoring and helping other people to, not telling people what to do, but helping you to learn to be good scientists and come from yourself.
That's wonderful. And before we wrap up the whole conversation, I have 3 quick questions for you.
Whoa.
It will be a rapid fire.
Okay.
You just have to give me your instinctive answers for those questions. Okay? Are you ready?
I'm ready.
So, in one phrase, tell me, what does the global safety science ecosystem need most to accelerate the NAM adoption?
It needs people to stop being fixed on what they've always been doing. They need to open their mind, and actually I think we need to remove ethics from a discussion. What we want to do is ethics is personal.
Yeah.
Everybody has their personal belief. It's ingrained in us.
Exactly.
Every time we talk about ethics, we're going to end up fighting with each other.
Yeah.
What we need to do is talk about science, because when we're scientists, we look at the data and we understand. That's what we need. That wasn't one line.
Exactly. It was in the one phrase, I would say. So, in one word, tell me, what do you expect from 3DCC 2026 conference?
Chai.
Really?
Absolutely.
Wonderful. Complete this. Human relevant models will redefine drug development by
Much better reflecting those diseases of the efficacy and toxicity than we've ever done before.
Oh.
We will reflect what we're trying to. We will be predicting disease. We will be predicting toxicity. Highly relevant to humans and for veterinary pharmaceuticals.
Yes, exactly. And wonderful. Thank you so much for these insightful words.
Thank you very
Thank
much.
you so much for having this conversation. Thank you so much.